All right, microscopy is actually what? Microscopy is the way through which we increase the apparent size of an object. Increasing the apparent size of a cell or any object, right, or any specimen. So that is called what? Microscopy. Why we are doing this? Because our eye having limitation. Below that we can see a thing up to that limit. Below that limit we can't see the thing well, like for example bacteria and other cells, human cell, right? So that is very small, so we can't see that. For that reason, in order to study these cells, we need microscopy. Microscopy, what we are doing, we are actually increasing the apparent size of any object, what we are studying. If that is cell, if this is organism, right? If this is the part of an organism, right? Like tissue. So for that reason, we need what? Microscope. Right? In microscopy, we use instrument called what? Microscope. Through microscope, we are actually increasing the apparent size of an object. For example, this is this size, and when you do microscopy for that, so you will see that large. Okay, now in microscopy, two things are very important. One is magnification. Another we have resolution. Okay. Magnification is actually the ability of a microscope that how much time it increase the number of the apparent size of an object. So the ability of microscope to increase the apparent size of an object. That is called what? Magnification. It means that how much number of time, right? For example, this is a thing. So how much greater this thing is that from original. For example, initially I will count it to 1 mm or 1 micrometer. And when after microscopy and microscope, when I check that the size of that increase, let's suppose it become 20 micrometer. Or like you can check it here, it become, for example, if it is 1 mm, right? And that become 40 mm, right? You can see that. So it means that approximately this thing increase 40 times. 4x, we mentioned it, x40, right? So 40 x 40 means this image increase, the size of this image we increase it, the apparent size, not the actual size. That increase 40 times larger than that of original size. So that is called what? Magnification. Okay, while resolution is actually the ability of microscope to distinguish between two closely present objects. For example, we have these two closely present things, object, right? Or cells, whatever this is. Now, the ability of microscope to distinguish between these two things, like to make it more clear for me, right? To make it more clear for me, not to enlarge that, but to make it more clear, it provides us more detail of a thing. Like it make the image more clear, not blurry. This is just like, you know, in mobile camera, when you are using that, if it is if the mobile having a good resolution power, so you can see that image more clear. That's not blurry. Is it okay? Actually, the ability of microscope, right? The ability of microscope to distinguish between two closely object. Between two objects that are very close. If the resolution power of microscope is more, it means it will provide us more detail of that object. You will see that clearly. It will provide us more detail of it. Okay, so there is difference between magnification and resolution. Magnification is actually the ability of microscope to enlarge the apparent side of an object, that how much time it is more than that of original. While resolution is actually the ability of microscope to distinguish between two closely, this is one point and this is another point. And when you check through one microscope, the image is blurry. While through another, when check it, you can see these 2 points very clearly. Right? Or if the microscope is not much good, you may see these 2 points is a 1 point. So it means the ability of the resolution power of that microscope is less. Okay? All right. So boys, here in microscopy, this is a bit large unit. So instead of this if we mention any other unit like micrometer, right, or nanometer. So for example, the object size we have, for example, 20 micrometer, and after microscopy, when I check that, right, it's become 100 micrometer, right? So it means the image increase how many times? Approximately 5 times is that of original. So here I should put it x5, right? X5 means 5 times magnification. Okay. Now it's depend on that. It can be like, you know, increase, or it's how it's depend on the magnification power of that microscope. Units of measurement in microscopy, you know, like we are studying very small thing to microscope, so the unit should be small, right? For the length, according to SI system, we have meter, but from that meter, other unit can be created if you are taking the multiple of 1000. If you are increasing or decreasing that. So let's move toward that, and we are now considering three main units that we should know about that. Millimeter, mm, okay, micrometer, and nanometer. Okay, now millimeter is actually 1,000 of meter. Which fraction of meter? 1,000. One-thousandth of meter. Is it okay? That is equal to how much? That is equal to 10, I think, right? While micrometer is equal to 1,000,000th of meter. That is equal to what? 10 by 6, right? And nanometer is equal to 1,000 millionth of meter. It means millimeter, 1,000th of meter. If you, for example, if it is a meter, right, and you divide this meter to 1,000 parts, so the last part, the last small part is equal to what? 1 mm, right? So for microscopy, we should know about this, that how to convert mm into micrometer and micrometer to nanometer. So usually these two units, micrometer and nanometer, we are using in microscopy, and we have to show our answer in these two units. So we have to know about this conversion, because sometimes they given us the data in, like two different types of data, and given us in millimeter, and they are asking answer from us in micrometer, and sometimes they provide us the data, like one kind of data they provide us in millimeter, another they provide us in micrometer. So for that, in that case, you have to convert that into same unit. Is it okay? So right now, let's see. For example, if we are converting millimeter into micrometer, what we have to do, and when we are converting micrometer into nanometer, so how we will do that? If you are converting micrometer into a millimeter into micrometer, so what you have to do, can you tell me? So in this case, we have to multiply this with 1,000. Okay, so that value will convert into what? That value will convert into micrometer. Now, if you want to convert micrometer into nanometer, in that case, you also have to multiply with 1,000. Is it okay? But sometimes they are asking a question from us that convert this mm directly into nanometer, so you should be careful about that. Millimeter into nanometer directly, so then you have to put 1,000, right? And again 1,000. Is it okay? So you have to multiply this value by 2 times 1,000. So it will give you nanometer. Am I clear up to here? Right? So this is the conversion of units. Now, in the same way, if you want to convert nanometer into micrometer, so now you are going reverse. Now you are going from smaller to larger. So at this time when you are going reverse, So this here, when you are converting micrometer into nanometer, you multiply it with 1000. So in this case, you have to divide that nanometer by 1000. Then it will give you answer. You will go reverse and you have to divide that. Okay, I hope up to here this is clear. Let's suppose the value given to me is 20 micrometer, right? And they are asking from me to convert it into nanometer. So what I should do at that time? I will multiply 20 with 1000, right? So it will give me what value? 1, 2, 3, right? 1, 2, 3, unit 10, 100,000, right? 20,000. What? Nanometer. Right? Same way, if you are converting 20 millimeter into micrometer, right? So what you have to do? You have to multiply that with 1000. With 1000. So 20 multiply 1000, it will give you the value per millimeter. Is it okay? Am I clear up to here? Right. Here mention here 10 nanometer. Okay, so 1 nanometer is equal to 10 minus 9. Okay. Boys, up to here, we learn about the units that are related with microscopy. Now we can do magnification. Magnification is actually the ability of microscope, how much time it is increasing, the how many time microscope is, or how many time it can enlarge the apparent size of an object. Okay, good. Now magnification, formula for magnification we have observed size of image divided by actual size of image. Right? Observed size of image means I, we are representing it by I and this one by A. Right? It's mean magnification is equal to what? IA. Is it clear? Now observed size of the image means that you check through microscope, that you see through microscope, right? This is observed size of the image. So yeah, image. While the actual size is actually, you know, like for example, it is small, right? This is the actual. And when you observe to microscope and you check, that is now big, right? For example, it is 20 micrometer, and after magnification it become 100 micrometer. So in this case, observed size is this one, while this one is what? The actual size, right? Now in paper 6 you was doing like this, that for example, this image is given to you, right? And then you draw that, correct? And you are drawing, you mentioned that large, right? When you measure this cell and then you measure this cell, or whatever the drawing or whatever. So if for example it with me is 40 millimeter, right? This with me is 40 millimeter. You can see here, for example, we have this 20 millimeter, this one, this. And when you draw your drawing, you mentioned that 50 mm. So in this case, this 20 is actually what? This 20 is real. This 20 is, no, this 20 is actual, A. So you will mention what? 20 down, right? and 50 mm you will mention up, right? Because this is you check after observation or after drawing your large drawing, right? So this is observe one, 50, right? Then you have to divide 50 by 20, right? It will give you the answer, and if you will write in front of that your answer. X time that you will put x mean how many times. Is it clear? So hope up to here you understood. Inshallah, we will continue our lecture from here tomorrow. All right, students. So let's move towards magnification once again. Right? The ability of microscope, how many times it will increase the size of an object. Formula of magnification, we wrote already. Formula of magnification, we have observed size of the image divided by actual. Right? You can also, from this you can also understand, just try to keep it. This is a very simple way. Sometimes, like if I'm finding magnification, so it is easy for me. Like for example, I will just put it here, observed size of the image, and dive it down, I will put actual size, right? But sometimes they given us magnification and actual, and they want from us observe, right? Or sometimes they given us observe and magnification, and they want from us actual. So for that, at that time, we have to deal with this formula and we'll convert it to different form. So like IMA, this is very easy way. For example, right, you are going to find for observe, so definitely you will do what you will magnification multiply by actual, so that will give you that value. This is very simple. For example, they asking from me about the observed size of the image, so you have to use maths rules. So this A will go there, right or no? So yes, obviously whatever I'm doing, right? So I will modify my formula, this both will cut, right? That multiplication will happen here, right? So it means observe is equal to A multiply M. Observed size of the image is equal to magnification multiply actual. Am I clear up to here? The same if you are doing for the actual, so observe divided by magnification, right? So by using this formula, you can, like if two value given to you, another you can find it to this formula, right? Now let's suppose I'm giving you example that let's suppose it's given to me here the observed size and actual, right? And they are asking from you the magnification. So I will use M is equal to IA, right? Now put the value here. Which value is given to me here? First of all, the value that's given to me for image size or observed size is 1/6. It's given to me 60 mm. Let's suppose 60 mm is given to me observed size of the image. It means I given to me 60 mm, right? While the actual given to me 80 micrometer. Now what they are asking from me, magnification. So whenever you are putting your value and formula, both should be in same unit. For microscopy, we are trying to convert the large unit into small. So in this case, we will convert that 60 into micrometer, right? So how we will convert? As we studied before, that whenever we are converting millimeter into micrometer, We have to multiply that with 1000. So it will give me approximately 0.0000 micrometer, right? Now 80 is already in micrometer, so I will put my value here. This 60 is equal to this 60,000 micrometer. I will put this value in formula now. Put that in formula. I 60,000 micrometer, observed size of the image, right? And the actual we have 80 micrometer. Now let's check how many times this microscope magnified our image. So when you divide this value by 80, can you do it? It will give me the value of what? 750. It means the image that microscope, the size, the apparent size of that image increased by or enlarged by that microscope 750 times. Okay, X show you how many times. Is it clear? So same you can do it for, if they given to you, for example, they given to you magnification, or if they given to you, let's suppose they given to you magnification, right? And they given to you actual. So in this case, magnification, for example, we have what? X750, let's suppose. They given to me this value. And they also given to me what? Actual. Actual we have 80 micrometer. So in that case, you will use this formula. It means that I will multiply 750, right, with 80 micrometer. That will give me I value, observed size of that image. Is it okay? All right, so now we know already, we discussed about that, human eye can, there is a limitation for human eye, like approximately 0.1 mm object size, right? If object size is decreasing than this limit, so human eye can't see that, right? So it depends on a person, like 0.1, 0.8, it depends, like, but usually 0.1, if it is decreasing, then we, like, a person can't see the thing, right? So for this limitation, we will face problem if we are dealing with such small things that are less than that. So what we have to do about that? Right? So definitely we will move toward microscopy, the way through which we are increasing the apparent size of an object. Apparent means not the real, but it seemed to us like that. Okay. Now, in this case, we will move toward microscopy, and if the first option with us is in microscopy is light microscope. Microscopy definitely we will do by microscope, right? So the two microscopes we have in this syllabus, the first one we have light microscope. Another one we have electron microscope. Okay. Electron microscope, we have two types: transmission electron microscope, electron microscope. Okay. TEM, simply we are STM, right? And scanning electron microscope. Scanning electron microscope. Okay, now this light microscope here, actually visible light will use. We will use light and we will study our specimen, the thing that we want to study through microscope. While in electron microscope, beam of electron will use. So the first one, boys, I'm considering light microscope, right? And then I will go toward electron microscope. First, light microscope. As I discussed with you, light microscope work with light, through visible light. And you know, the visible light is moving in waves. They are moving in waves, right? Like in this way. Now, if this wave is, you know, if it is falling on any object, you can see that. So the visible light range that human eye can detect, right? So that is 400, that human eye can see 400 to 700 nanometers. You can check here in this electromagnetic spectrum, you can check here, we having different range of wavelength, right? Each rays are each, you can see here, X-rays, UV light, infrared, all having different wavelength, right? Now, visible light is 400 nanometer to 700 nanometer. Okay. In this range, you know, if this range, this wavelength of light, we can detect it. Our eye can detect it and send to brain to convert into color. Right? So it means in this range we can see it. So here is a general formula for this. That, for example, I divide the low visible range, that with me is what? 400 nanometer by 2. So this will give me value 200 nanometer. The low range that you divide it by 2, the low visible range, and it gives you 200 nanometer. So through light microscope, we can see only object that is small up to 200 nanometer. Am I clear? So it means that the resolution power of this light microscope is 200 nanometer. Right? This is just general formula that the low visible range, so the low is that when you divide that wavelength by 2, so it will give you 200 nanometer. This is the resolution power of what? Light microscope. While the magnification power for light microscope is approximately 500, right? And 1500 or 2000, it depends on the microscope, because new microscope is coming to the market gradually, right? So up to here, I hope this is clear. Now, you know, if the object with us is, for example, that is decreasing from this, that is decreasing from 200 nanometer, then we will also face problem. Okay, then we will face problem, and for that reason, you know, we can't see that through light microscope. Then we will move toward electron microscope. But first check what is light microscope. If you see here, In my hand, this is a light microscope, you can see, right? You know, in this place, let me discuss with you. First of all, I will show you here. You can check here in this picture. First of all, boys, check on my screen. You can see here this part of microscope. In this part of microscope, we can put our specimen, right? This part you can see stage. In this stage, we will put our slide, right? This is stage. You can check here. This is stage. And that we will put our slide, right? In this place, this is light source. Light will fall on this specimen. What is in this light will fall on it. And then, you know, it will pass through this objective lens. In this place, we having a lens that will modify that glass lens. That will increase the size of that image. While this is eyepiece, okay, this eyepiece, with near to this eyepiece, we have also another lens called ocular lens. It's not mentioned here that it's called ocular. So to this eyepiece, we are looking to what? To the image which we are interested, to the specimen that we want to study, right? And this one is, you can see here, this one is arm of this microscope. While the important thing for us is these two things. One is coarse adjustment, another is fine adjustment. This one is, you know, if you check here, this coarse adjustment is, you can see here in the screen, this big button, this rotator. This big one is called coarse adjustment. While if you see with coarse adjustment, there is another on this up on the coarse adjustment, we have this fine adjustment, you can see. So when, for example, like if I put my hand like this, so this one is coarse adjustment, this one, right? This coarse, and this one we have fine adjustment. Okay, and through this button, you can put your stage here and there so that you adjust it. Practically, I will do this to you, don't worry. Okay, so in this case, what will remember? Coarse adjustment we are using to check image, to check, sorry, specimen. For example, first of all, when we put our slide, so coarse adjustment, we want to bring where is the specimen. It will give us image blurry. We will find through that stage, will go up and down. But when we find our place of specimen, that image will be blurry. So to make it more clear, to bring it to our eye range, because my eye range in other person may be different. So for that reason, I will do fine adjustment. A bit I will go like through this button up so that it make me more clear the image. Okay, so this is about light microscope. What is happening in light microscope? Light will pass through the specimen, right? And then objective lens, here is the lens that will magnify that, and then that light will fall where? That will, when it falls through the specimen, definitely that specimen image will enter to the lens, and that will make it magnify, and then final magnification will do by ocular lens. Here is another lens with this eyepiece. And this eyepiece is the region through which we look to the thing, the specimen. Okay? Is it clear? And this region is base. Okay? This is working with simple light bulb is here in this one. I hope up to here this is clear. Now, let's suppose, boys, like how through this light we are detecting an object. Actually, you know, like this light travel in waves, right? If object is here, And front of that, for example, this thing came, right? So it means this object interfere with that light. That's why we can see that. That's why we can see that because of that interference. Am I clear up to here? So then your lens can, if definitely it will block that light, so that place will magnify by your lens. Now, let's suppose this is our light and the minimum wavelength of that is approximately the minimum we have. In this case, we have 400 nanometer, right? And here I take it mitochondria. Mitochondria, that size is approximately 1000 nanometer, right? So it means this mitochondria can interfere with that light, and that mitochondria can block that wavelength, and we can see that, right? Now, let's suppose if I have another organelle, that is this, right? For example, I have this another organelle. Size of that organelle, I have 25 nanometer, and the name of that organelle is ribosome. So if I put, like, for example, I'm passing that wavelength of light, again, visible range, right? So this wavelength may not touch that. It may escape. Means that wavelength can't touch it. This cannot interfere with this wavelength. It will remain in the middle. That's why I can't detect that. I can't see through visible, very small I cannot see. So for me, now the solution for this is that I should have such thing whose wavelength should be small, like this, more energized, so that it detects that this ribosome can interfere. If wavelength is small, so definitely that can ribosome or any other small thing interfere. So for that reason, I have to move toward another options. With me, another option, according to my spectrum, according to this spectrum with me, other options that having small wavelength or less wavelength than that of visible light, that is X-ray and ultraviolets. But the problem is that I can't use this because scientists face problem in focusing of these rays. They were facing some problem in focusing of these rays, right? Because if you check here, X-ray, UV rays, it decreased, like the wavelength of that is less is comparatively. This see that this show you, so this down show you what here. If you check here, this one, let me take it. This one, sorry, I will take another. Yes, this wavelength, this is shorter, even gamma, like if you are going to that side, the wavelength becomes short. So through this wavelength, I can solve my problem, but scientists facing problem in these, to focus on that, to focus it on the specimen. So for that reason, we will move toward electron, because electron, if we bombard electron having very short wavelength, right, very energized, if we bombard that electron, so we can check that the small thing can also interfere in the way of that, and we can see that. We can control electron when we are passing that through electric magnetic field, right? So we can focus it in a specific point that we want to do by passing beam of electron and electromagnetic field. Okay, so for that reason, we will move towards electron microscope. Through that, we can resolve a thing like, that we can see a thing about 0.5 nanometer. That is the resolution power of microscope, electron microscope. Am I clear up to here? All right, students, in the last lecture, we discussed about light microscope. So today we are going to discuss about electron microscope, okay, in detail, and the difference between light microscope and electron microscope. As we discussed already, that the resolution power of light microscope is 200 nanometer. Okay, the general formula was that the wavelength of visible light you divide by 2, it will give you that up to that limit you can check, you can study that specimen, up to that size you can study it. For example, if you are using 500 visible light, so you have to divide it by 2, so it will give you 250 nanometer. So up to that range, by that wavelength, you can study any specimen. Okay, now today we have light microscope, electron microscope, electron. As we discussed, that an electron microscope beam of electron will use. Why we are using here electron? The first thing is that electron having short wavelength. If it is short wavelength, so it's mean a very small thing, like is I given you example of ribosome that can interfere on the way of that electron. Okay, so for that short wavelength, we are using beam of electron. And why we are using electron? One is short wavelength, okay, and another we have they can block short wavelength, okay. We can focus that on specimen by passing through electromagnetic field. Okay, now if you see here, electric magnetic field, through that we can the beam of electron we can pass through electromagnetic field, and that can focus electron on specific spot. Okay, now the first thing in electron microscope is electron beam of electron well, electron gun that will fire what? Beam electron gun. This one you can see here, this one electron gun, electron will be fired from here, and that will pass through condensers, right? This is actually electric magnetic field, and that is focusing that project that is focusing on the specimen, right? So you can put your specimen here. For example, this is your specimen here, right? So when electron beam pass through that, okay, so it's the edge, it will strike a fluorescent screen, right? So the place where that fluorescent screen is black color, so the place where it struck that fluorescent screen, so that place will be white. Am I clear? The place, for example, it is a black color sheet, right? And definitely if the specimen is coming on the way of that electron, so that will block that electron. And the place, and if the specimen is not interfering electron, so that place it will struck by that electron. So the place that's struck by electron, it will become white, while the remaining will remain black. So you will see black and white image will form on this fluorescent screen. Okay, then we analyze, then we study that fluorescent screen through different software, and through computer screen we can study it. But we will not move to we will not go in detail in that, but just keep it in mind that electron gun will fire electron that will pass through condenser lens, right? That will pass through even magnetic field, right? And that will focus electron on the specimen. Okay, when it's on the way specimen is coming, that will block the way of that electron. Okay, so on the fluorescence screen, that place will be black while the remaining one will remain white. So black and white image you will take it from here. Fluorescence screen will be black and white, and later on you can analyze it through different software, and you can study through computer screen. Okay, now here we are taking all this when we are bombarding this electron. So the place should be, this should be vacuum. All the area, it will be vacuum. Vacuum means there should be no air, nothing. Okay, why? So that may not interfere on the way of electron. Okay, another thing, electron microscope when we are using, usually not, within electron microscope we can use this for the dead specimen, not for live. The reason is that inside that the temperature is high, and because of that evaporation of water can happen from that specimen, and that's because if it remains for a long time, dehydration will happen and that will become dead. So for that reason we can do only this for dead specimen, not for live. Like we can't do this for live bacteria and other live cell. Okay, is it clear now? But the good thing for electron microscope is the resolution power of electron microscope is very like very high, we can say, up to 0.5 nanometer. It can, if the size is up to 0.5 nanometer, we can see that through electron microscope. If there was a limitation with the light microscope, if the specimen size decreased from 200 nanometers, so we can't see that. But in electron microscope, we can see up to 0.5 nanometer. Okay, now there are two types of electron microscope. One is transmission electron microscope, okay, and another is scanning electron microscope. Okay, transmission electron microscope used to, if you want to study internal structure of a cell or any specimen internal. Okay, while scanning just show you the three-dimensional image and only external appearance of a thing. Like it give you the external appearance in more detail. While transmission electron microscope, it will give you internal detail of the specimen. Okay. Now, in light microscope, you can see here, so the advantage of light microscope is that we can study a live specimen to that, because only light is passing through that. If the specimen come on the way of that light, you will see that. Okay, you can see that through your eyepiece. Okay, you are using here, you are looking the thing through eyepiece, the specimen, while in electron microscope you are using the screen. Okay. Now, here we are using glass, here we are using glass lens, and there we are passing that to electric magnetic field, okay, condenser lens, okay. And another thing for light microscope is, light microscope is not expensive, while electron microscopy is expensive, in that need more technical training as compared to light microscope. Okay, so this is the advantages, disadvantages of both. You can see here that this is lamp. You can check here the microscope from downside. Light will pass through lens, and that after that it will pass through the specimen, right? Specimen, and by passing through the specimen, for example, here this is a specimen that will pass through objective lens. Okay. And when it pass through objective lens, so what will happen? That will pass through eyepiece, okay, eyepiece lens, okay, and then you can see that through eyepiece. Ocular lens, also called ocular lens. So you can check final image, you can see by through eye, okay. But here you can see that its image will form on fluorescent screen, and that fluorescent screen you can see, check through computer screen. Is it okay? I hope up to here this is clear. So only here you just remember glass lens we are using, and here electromagnetic lens called condenser lens. Okay. Any question about it? Do you have about this topic? You can ask. Boys, you know that like this is lamp and the light microscope. So this is condenser lens. This condenser lens actually, you know that the light is in spread form. So this condenser lens is near to the lamp, okay, to the light source, and that focus that light on your specimen. And after that, objective and ocular lens will magnify that. Okay. Now, usually remember, just remember this point. For example, we have one eyepiece. Remember, boys, this is, for example, this is your eyepiece, okay, through which eyepiece the lens that is here, okay, with eyepiece, okay, so that is ocular lens. So you will check here within eyepiece magnification update is mentioned. Update lens magnification is mentioned. The eyepiece lens magnification mentioned on that. Like for example, here mentioned to me X40. Am I clear? This is very important point, X40. Now, objective lens that's near to this lens, I'm talking about the objective lens. Objective lens, the one that is near to the specimen. This one. Many objective lens we have, right, at different on, like for example, this objective lens is 40x. This one, let's suppose this is 100x. And let's suppose this is 20x. Okay. Now you can rotate this lens. For example, if you want to study your, image or your specimen through this 40x, you can rotate it in microscope, right? So you will put this 40x light. If you want to study through 100x, you can rotate it. So let's suppose my eyepiece is 40x. When I check it, it is written on that eyepiece already. Every eyepiece it's mentioned. In my hand here is also one eyepiece. You can see here on that magnification mentioned to me, please. One eyepiece is now in my hand. I will show you now. This one. In this magnification is mentioned already. You can check here. You can see, right? This. So how much magnification is mentioned on this eyepiece? 16x, right? So for example, I am studying an image with eyepiece of 16x, and down objective lens I keep it 100. Okay. So let's suppose if I'm studying my image, I have 16x right now here. Okay, and the down objective lens I keep it what? 100x. So x should be in front, sorry, x16. Okay, this is my eyepiece. Okay, and the another one I have x100. That is my objective lens. So now when I studied that specimen, so what I can conclude that how many times my specimen size increase. I'm talking about magnification. So in that time, you will multiply 16 with 100. 16 with 100. If you multiply it 16 with 100, so it will give you 1600. It means x 1600. It means my specimen enlarged, the parent size of my specimen enlarged by this eyepiece, and the objective lens that I keep it 100. So 16x means 1600 times that specimen size increased. Okay. Now let's suppose if I'm using this eyepiece again, x16, right, and I'm using my objective lens 40, x40. It's written already on objective lens like this. So what should be the magnification of that? Again, you will multiply 16 with 40. So it will give you the answer of that. That's how many times it magnified the image. Okay, I hope this is clear.